So we're going to start off with Farhan Augustine, who's currently a grad student at University of Maryland and is going to present his work that he's been doing at Johns Hopkins under Harvey Singer. Go ahead and take it away, Farhan.
Hello, everyone. Are you able to see my screen yet?
Yeah, you're good.
Yes, okay. So my name is Farhan Augustine. I am a clinical research assistant at Johns Hopkins School of Medicine, and I am a graduate student pursuing a PhD in biological sciences. And I will be talking today about my work with deer mice. The picture here on the right is of a deer mouse. I found this on internet under Creative Commons license. And my talk will be primarily centered around the cortical endogenous opioid findings and the link between repetitive behaviors. So to begin, let me introduce deer mouse and the type of behavior that we observe in these mice. At an early age, mice begin to display frequent repetitive behaviors, such as the ones you're seeing here: repetitive jumping, back flipping, pattern running, etc. And what we hope to do is to understand the pathobiology of these behaviors and to draw parallels between human clinical disorders such as OCD, tics, motor stereotypies, and autism, and try to understand from an animal model perspective how we can apply the information from animals to the clinical disorders. Behaviors are often talked about in terms of being goal-directed and habitual. Goal-directed behaviors are consciously performed, they are controlled, flexible, and adapt quickly to whatever the desired purpose is. The pathways are thought to originate from prefrontal cortex and the orbitofrontal cortex and project down to dorsomedial striatum. On the other side, the habitual behaviors are performed as repetitive, they're stimulus driven, they're slow to change, and often have no or little link to current or future goals. And the pathways that are considered habitual pathway originate from premotor cortex and supplementary motor area and are thought to project to dorsolateral striatum. I have included ventral striatum in this figure because recent studies are beginning to show that ventral striatum may be perhaps gating the transitions between goal and habitual behaviors. The other major pathways that are thought and talked about in literature is this direct and indirect pathway system. Motor information from the cortex is projected down to striatum. In the striatum, genotypically distinct medium spiny neurons of the striatum that either express dopamine receptor D1 or dopamine receptor D2 are thought to then segment that information into direct pathway or the indirect pathway. The direct pathway MSNs express dopamine receptor type 1. The indirect pathway MSNs express dopamine receptor type 2. And the flow of information goes from striatum to the globus pallidus interna via the direct pathway, D1 expressing MSNs. And that is thought to enhance the motor activity, whereas the indirect pathway going to the globus pallidus externa is thought to diminish or reduce the motor activity. However, in reality, the situation is a lot more complex. There are a lot more interactions between different regions and different neurotransmitter systems than we talk about when we just talk about behaviors. So here's a picture from one of my publications from this year that depicts or tries to depict how many different interactions there are between different regions of the motor circuit. The one on the right is just a simplistic representation of the one on the left. It depicts just the bidirectional or unidirectionality of these connections and interactions between different regions. So please keep in mind that interactions and connections are very complex. Researchers all over the world are trying to understand the best that they can. However, recognizing the multiple interactions between the neurotransmitter systems and the different regions, the question is, where do you start to look for the problem of repetitive behaviors? The problem could be anywhere in any of these interactive pathways. So when I joined the lab and I took the deer mouse project on, I decided to focus on the cortex, striatum, and basically the basal ganglia. Basal ganglia in the mouse kind of looks like this. And what I did was I took a neurochemical approach to try to identify if there were major neurotransmitter abnormalities that we could identify, and that if we can correlate those abnormalities to the repetitive behavior, then we could potentially have a hook that we can use to pull on the string of this repetitive behavior pathobiology. So I chose to look at 5 of these neurotransmitters: glutamate, GABA, dopamine. Dopac is actually a metabolite of dopamine, and the number 4 and number 5 are both opioids, leuenkephalin and dynorphin A. The rationale for measuring opioids is multifactorial, including their ability to regulate the release, presynaptic release of glutamate, GABA, dopamine, etc. And endogenous opioids also play a major role in the habitual and goal-directed pathway systems, as well as the gating of the striatal direct and indirect pathway systems. So from the findings of the neurotransmitters, what we found was that in the FC, in the frontal cortex, GABA neurotransmitter was reduced compared to the prefrontal region. And the leuenkephalin levels were higher in the frontal cortex compared to the prefrontal cortex. And it was statistically significant difference between the two regions when it came to opioid enkephalin levels. Luckily enough, the enkephalin and dynorphin levels both significantly correlated with the repetitive behaviors. So the lower the enkephalin level was, the greater the activity we saw. So keeping all of these things in mind, we came up with, we tried to understand what might be going on. So from doing some literature research, I found that GABA interneurons are primarily expressing enkephalin in the frontal, in the cortical regions. Therefore, assuming that enkephalin, which is represented here by these stars, is inhibiting GABA, and GABA then disinhibits the cortical pyramidal glutamatergic neurons, which will fire down into the striatum, therefore innervating the dorsolateral striatum, because that is part of the habitual pathway system. I should reiterate that habitual pathway, the frontal cortical regions innervate the dorsolateral striatum, whereas the goal-directed pathway, the prefrontal cortical regions, innervate the dorsomedial striatum. So keeping our data in mind that we have found higher levels of enkephalin and lower levels of GABA, again represented here by these stars and the broken red arrow of this GABA, what we are hypothesizing is more innervation of the dorsolateral striatum. Excuse me. Okay, then what about the direct and indirect pathway system? In literature, I found that prefrontal pyramidal neurons primarily innervate D2 expressing MSNs, which are part of the indirect pathway. So keeping that in mind, if the pyramidal neurons are disinhibited in the frontal cortex, we should expect greater innervation of the D2 expressing MSNs. or perhaps even D2 expression itself, the receptor expression itself. So what we then decided to do was we decided let's overcome the LCMS analysis, some of the challenges that we find in the LCMS, the liquid chromatography mass spectrometry, is that we lose the spatial context. We lose the ability to see where in what region are we exactly. So therefore, we decided to go with in situ mRNA hybridization, where we can have a greater control and we can see in what regions are we spatially, and to understand the expression level of different receptor types in the frontal region and the prefrontal region and the striatum. Some of the things we are currently quantifying are the D1, D2 receptor expression, as well as mu delta kappa opioid receptor expression in the cortex and the striatum. I have some preliminary data from the current directions and the current investigations. So again, the hypothesis that we came up with from the liquid chromatography mass spectrometry was that we would observe a greater innervation of dorsolateral striatum via the disinhibition of glutamatergic pyramidal neurons in the frontal cortex. And another way to state this is to say, if cortical neurons in the FC are less inhibited and there is less production of opioids in the dorsolateral striatum down here, which suppresses inputs from the frontal cortex of mice that display high activity, then we should expect to see an inverse or negative correlation between the opioids that are released here and the activity of the deer mice. So the preliminary analysis from the RNAscope and the expression of dopamine and the opioid receptors is showing some promising results that support the hypothesis. However, there are a lot more questions that are generated from the data. What we have observed so far is that in the dorsomedial and dorsolateral striatum, the dopamine receptor expression positively correlates with the activity of the deer mice. And the same thing is happening with the ventral striatum, ventromedial and ventrolateral striatum. However, the correlations are more significant in the lateral part of the striatum, here shown up here and down here. So these are the two regions that are involved in the habitual pathway. As we had hypothesized, the dorsolateral striatum shows a significant positive correlation between the dopamine receptor expression as quantified by the in situ RNA hybridization and the repetitive activity. However, it generates more questions because D1 receptor expression is thought to lead to more motor output from the striatum. D1 MSNs are involved in direct pathway, therefore it is believed that direct pathway leads to more activity. However, what we are finding is that perhaps there are more D2 expression, there's more D2 expression in the striatum than we previously thought, and perhaps that is one of the things we should further look into. Here's an expert opinion or excerpt from a recent paper I was reading, which suggested that D2 MSNs inhibit direct behavior, and it leads to goal-directed behavior. Whereas the ventrolateral striatum D2 MSN activation initiates the action, and the fact that that we are seeing the ventrolateral D2 expression and positive correlation, a significant positive correlation with the activity, suggests to me that D2 MSNs, D2 expressing MSNs, are perhaps more involved with our repetitive behavior phenotype than we previously thought. The comparison I'm trying to draw here with these two graphs is a meaningful one, because in the striatum, D2 expressing MSNs are the ones that release this Lewin-Kaplan opioid neurotransmitter. And as the expression of dopamine D2 receptor goes up, we are seeing the expression of opioid and Kaplan going down. And what this is indicating to me is that we need to take a deeper look at the expression level as well as the activity level, and how we can do that is through electrophysiological recordings. We can identify MSNs based on the dopamine D1 expression and D2 expression, and do electrophysiological recordings. We can perhaps even do pharmacological manipulations and see the behavior impact of, let's say, introducing a opioid antagonist and agonist in a specific location in the dorsolateral striatum. Or perhaps there are other regions that we should explore that may be introducing situations that are almost significant, such as the D2 expression in the dorsomedial striatum, that could be coming from regions like thalamus or hippocampus or other areas of the brain. So by no means do we have exact answers, but we are a lot more closer to understanding the role of endogenous opioids and their gating of the cortical output to the striatum than we were previously.
Sorry, Farhan, I was so wrapped up in what you were saying that we've run out of time.
Oh, wow, it's been 15 minutes.
I'm sorry. We have a question for you. If you want to type to that, that'd be perfect. Or we can circle back at the end if we have time. We're going to quickly switch gears. Sorry, I was so